Hello there, and thanks for tuning in to see what the world looks like through atheist eyes. I'm Frank Zindler from American Atheist Press, and the atheist eyes through which you are invited to see the world are my eyes. Today, I wish to present part 3 of a series of programs that I've titled Immortality Now, Living as Long as You Wish. This part of the series is subtitled From Mayflies to Methuselahs, and like the first 2 programs in this series, it is a video adaptation of the first chapter in the 2nd volume of my Red Books. My Red Books, a 4-volume collection of my shorter writings, are titled Through Atheist Eyes, Scenes from a World That Won't Reason. The first chapter of Volume 2 is titled The Prospects for Physical Immortality. Come on, let's take a look at what you need to know if you want to be a Methuselah instead of a Mayfly. In the first program of this series, I told you that science is making extraordinary advances in understanding the causes of aging and is close to understanding how to retard or stop the aging process. In fact, we probably aren't all that far away from understanding how to reverse the aging process right now. I told you that some of you now watching these programs have a good chance of living to be at least 150, even though it's probably a bit too late for me. In part 2, I explained the evolutionary background for the lifespans of various plants and animals, and I explained why religion, especially Christianity in general, and Catholicism in particular, has impeded all scientific attempts to understand the aging process and has prevented us from discovering the physiological fountain of youth. Priests and preachers won't be able to collect rent in advance for angel-feathered beds in heavenly mansions if you are going to be able to continue sleeping in your sleep number beds here on planet Earth. I suggested that we consider aging to be a disease, just like any other disease, and that we try to discover its causes and mechanisms. I suggested that there probably was a genetic reason for at least some aspects for aging, and today I want to consider that possibility. We don't have to look far to find a clue to genetic aspects of aging. Some things literally leap up to capture our attention. Consider salmon, for instance. In nature, salmon are hatched in freshwater streams. They develop through several stages, and then they descend to the sea to grow to sexual maturity. As much as 5 years after their hatching, the nubile fish reascend the rivers that gave them life in order to consummate their nuptials. How the fish recognize the natal waters to return to their watery nurseries is itself a subject worthy of an entire program. At this time, a most remarkable transformation occurs. The formerly sleek, silvery blue fish become a dull, reddish brown. The males, in particular, become ugly and misshapen. As the fish spawn, they grow suddenly old. Most die within a few days after the fertilization of the eggs. Rare indeed is the salmon that survives to spawn again. It is as though the fish had happened into a never sought for fountain of senility. Are there genes that are shutting the fish down? Indeed, there are, as we shall see shortly. Similar to the case of the salmon, though even more dramatic, is the case of the mayfly. These insects are classified in an order named ephemerida, in recognition of their ephemeral existence. Incidentally, the word ephemeral comes from Greek and literally means upon a day. All but 1 or 2 days of a mayfly's life are spent as aquatic larvae. At last, they emerge from the water, try out their newfound wings, perpetuate their race, and perish. Within hours, their temporary terrestrial tenure is at an end. And then there are carrots. Carrots, as everyone knew back in the days when I was going to a 2-room country school in Michigan, carrots are biennial plants. That is, they live exactly 2 years. Flower, go to seed, and die. They die not at the end of 1 year, not at the end of 3. Almost always, they self-destruct at the end of 2 years. A similar, but more impressive phenomenon is seen in the case of the so-called 17-year locust, actually a cicada. It's a cicada which lives for 16 years as a larva, emerges as an adult in its 17th year, mates, and then seeks out a recycling service for its carbon atoms. Is there any relationship between salmon, mayflies, cicadas, and carrots? I think so. The precision with which their aging and death occur makes one suspect that there is some sort of genetic predestination at work. It is as though evolution has equipped these species with self-destruct genes whose function is to get older generations out of the way in order to avoid competition with their descendants. Back in the late 70s, when I first started writing on this subject, no technical word for such self-destruct genes existed, and so I coined one. I termed them opsiphonial genes, making up a word from the Greek, opse, meaning late, and phonios, meaning murderer. Alas, the term is not caught on, even though, as we shall see, such genes have at least something to do with aging in all animal species. Are opsiphonial genes at work in human aging? While humans are neither annual nor biennial, they are, if I may be permitted the barbarism, they are proverbially 70 While improved care and nutrition have steadily increased human life expectancy, they have not significantly increased our life span. That is still nearly fixed at about 70 years, although some recent authors feel the human lifespan really to be about 110 years. The world record is held by a French lady named Jean Calment, who lived 122 years and 164 days. Even so, it appears that opsiphonial genes must be at work. Our suspicion is further strengthened by the bizarre disease known as Werner's Progeria, an outrageously premature development of old age. By the age of 8, a child may be biologically 80. It seems as though something has happened to activate prematurely a normally dormant opsiphonial gene or genes. Of course, the same effect might be achieved by the sudden turning off of a gene needed for some vital function or by a mutation in such a gene. But it seems not irrational to suppose, therefore, that if something can turn on such a gene prematurely, we should be able to find a way to turn off or at least delay such a gene's activity as well. Many people, when discussing the genetic aspect of aging with me, express the hope that aging will not prove to be determined genetically. they shrink from the notion as if it were a sort of biological Calvinism. What hope can we have if our very genes are against us, they ask. Actually, if suicide genes were the whole story, we would be in great luck, for we already know a great deal about what turns genes on and off. Hormones, for instance, are busy at this very moment turning on and off various of your genes as you watch this program. Other genes are being turned on or off as a result of the buildup or depletion of various simple chemicals in particular tissues of your body. Many of your genes are being turned on or off at this moment by particular proteins called transcription factors, located at strategic points in the nuclei of the cells of your body. Most exciting of all, genetic engineers have learned to read and write in genetic code. It is now a daily occurrence for human genes made of DNA to be combined with particular synthetic or natural sequences of DNA, which will guarantee expression, that is to say, turning on the gene in a new host cell, say a bacterium. Yes, it must come as a shock to creationists and other flat earthers that completely human genes can be transferred into bacteria and virtually any other organism and continue to function. Work of the devil. Well, maybe, but I hope they'll think about this whenever they treat their diabetes with human insulin produced in bacteria. Humans are not the products of special creation. Okay, back to opsophonial genes. That which genetic engineers turn on can be turned off as well. In any event, if aging and death were due to the action of suicide genes alone, the cure for old age would be conceptually no more complicated than finding the legendary fountain of youth. All we would need to do would be to find a drug or some form of magic bullet which was specific for the gene in question and was able either to repress it or permanently disarm it. We started this program with salmon, and I think that might be a logical subject with which to end it. You will recall that I opined that one or more genes must be at work to shut down the salmon as soon as they have spawned. As I was preparing this program, I was delighted to discover that the programmed aging of salmon has become the subject of a very energetic research program, actually programs. Salmon aging is yielding clues to the aging of humans and other animals as well. It turns out that the migration of salmon back from the ocean to the streams of their origin is a very stressful experience. As with the induction of stress in humans, the salmon's brain triggers the release of great amounts of cortisol from adrenocortical tissues. It is thought that this acts on the hippocampus in the fish's brain to help it recall the so-called home stream olfactory bouquet, to allow it to zero in on the exact stream in which it was hatched and began to mature. Unfortunately, the cortisol triggers a cascade of degenerative processes resulting from the activation or inactivation of many genes now being identified. It is well known that chronic stress accelerates the aging of men and women. Just think about how rapidly several recent presidents really began to look old before their terms were up. Indeed, it appears that chronically high levels of cortisol in humans is part of the reasons for this. Now, it would be nice if all we had to do were to reduce or cut off the effects of cortisol. But of course, doing that would probably kill us faster than anything else, faster than the chronic stress. Even so, the study of the genes involved in the accelerated aging induced by cortisol will be of great help in learning how to engineer and regulate the genes that determine our lifespans. Hopefully by now, you want to know more specifically what genes are opsophonial, late killers, and what is known about how they work. Luckily for you, a bunch of them now are known. In the next program, Part 4, we will consider the best known and the best understood ones. We will consider how their control might lead to immense increases in our lifespans. Appropriately, the next program will be titled Methuselahs' Genes. That's all for today. For American Atheists and for American Atheist Press, I'm Frank Zindler.